All right, lipids are a little more complicated. We know that carbohydrates are absorbed right into the blood. Proteins are absorbed right into the blood. Lipids, not. Okay, so lipids are not directly absorbed into the blood. They're actually absorbed into the lymphatic system. So they're going to be absorbed into specialized lymphatic vessels within the digestive tract called lacteals. So when you, well, let's look at lipids really quick. All right. So we know that lipids are obviously fat. They are hydrophobic. When they enter the body, when you eat fats, our body is a watery environment. So in the watery environment, of the small intestine, once it gets there, not much happens to it when you chew it up and swallow it into your stomach. Not much happens to it from there. Okay, there's a few little lipases that are in the stomach, but not a lot of fat digestion occurs. So it goes into the small intestine, and when it's in there, it's very watery in there, and it forms a large fat globule. So just like, a salad dressing, oil and vinegar, they separate, right? Well, fat is going to do that in the body. So it forms these big old fat globules, and the enzymes that break down fat are called lipases. And lipases are hydrophilic. Okay, so that means they are water soluble. So these water soluble enzymes, they basically cleave the ester bonds of the lipid, and that breaks down triglycerides into fatty acids and glycerol. So they break down triglycerides into fatty acids and glycerol. So because the lipase is hydrophilic or water soluble, it can only contact the outside of it. So keep in mind that the fat is all bunched up in this big old globule. So a fat globule. So the enzyme can only contact this outside here. Okay, it can't get in here, so it can't really do much. So in order to be able to break down that, enzymatically break down the fat, we have to break this thing up. Okay, so we have a special fluid in the body called bile. All right, and bile is used to break down those fat globules into smaller molecules. So it breaks down fat globules into smaller molecules. That is called emulsification. Emulsification. Okay, so bile is an emulsifier, and emulsification just means taking a big fat globule and breaking it apart into smaller little globules. And once we do that, then the surface area to that fat will be increased dramatically, and the lipases will be able to actually get on there and break things down. So bile contains amphipathic molecules. Bile salts are amphipathic. It contains bile contains phospholipids. It also contains cholesterol, and all of these things are kind of utilized when we are working on breaking down the fat. So if this is the fat globule here, let me just and when we add bile, it breaks down this fat globule into little globules, little fat molecules here. Okay. So again, it increases the surface area. Now we can get to all of these rather than just, what was outside here. Okay. So bile also contains some constituents in order for lipases to be able to do their things. So we know that lipases are water soluble. So the lipases kind of need a little help in order to be on there. So there is another enzyme called Lipase. So I'm just going to put that here. So Lipase or colipase, however you want to call it. And what it does is it helps the lipase, and I'll make that yellow, to bind on here. So here's the lipase, and it's going to bind onto these little globules of fat. But again, it needs sort of that little help here. Lipase is amphipathic, which means it's really not hydrophilic or hydrophobic. It can kind of be happy anywhere. So once the lipase is there, then it will take these emulsified particles of fat and break them into fatty acids and or into free fatty acids and monoglycerides. Okay, so we keep going here. Remember that bile has phospholipids in that, and so it takes those phospholipids and breaks this fat down into its fatty acids and monoglycerides, and it encases them into these things called micelles. And micelles are basically a fatty acid surrounded by phospholipids. They're these little particles. So a micelle can also encompass a monoglyceride as well. Okay, so once these fatty acids are formed, or these micelles are formed, now they can go into the small intestine cell. So if we have this is our enterocyte, or small intestine cell. Okay, put a little nucleus there. It's a cell. And the micelle, how does it get in? Well, it can go right in. these are phospholipids. These are phospholipids. So they're fat. And the plasma membrane is basically phospholipids, which is a fat, so they can go right in. When they get in, they go over to the smooth endoplasmic reticulum. So the smooth ER. And if you remember the smooth ER, that was where fats are kind of made and fats are formed and all of that. So those micelles that are holding fatty acids and monoglycerides go over to the smooth ER in the smooth intestine cells, and then they reassemble them into triglycerides. So once in the smooth ER, then fatty acids and monoglycerides are reassembled into triglycerides. Okay. Then from there, those triglycerides are going to be, we're going to take those triglycerides and actually put them into something that we can send them out and they can travel in. Okay, so the next video we're going to talk about how those triglycerides end up in the body or in the blood.